So we will be starting today a new chapter, osmosis, diffusion and active transport. Okay. So have you ever heard these terms before? What is osmosis, diffusion and active transport?
Diffusion is the spreading of the particles, right?
Mm-hmm. Osmosis and active transport.
I don't know about those.
Okay. So osmosis, diffusion and active transport, all of them are the movement of the molecules. And since we are doing biology, so these are actually the movement of the molecules in and out of the cells. In and out of the cells, movement of the molecules in and out of the cells. So I will show you the slides. Let me first explain what is this phenomenon actually. All right. So movement of the molecules. Can you see my whiteboard?
Yes.
Movement of the molecules in and out of the cells. Okay. So what are the three types of the movement? They are diffusion, osmosis, and active transport. Osmosis, diffusion, and active transport. So these are the three ways by which the molecules move in and out of the cells. So let's start with first diffusion. What is diffusion? Diffusion is, for example, this is a cell. All right. So in case of a cell, whatever the nutrients and oxygen molecules are there, if they are at a high concentration, if they are more out of the cell than in the cell cytoplasm, then simply they will move from high to low concentration like this. Whatever the waste particles are there inside the cells, if they are more inside the cell than the outside, they will simply move out of the cell from high to low concentration. What is the meaning of concentration? Abundance of the particles. So if the particles are more on one side and less on the other side, then the particles will simply, they will spread from the area where they are more to the area where they are less. So we say that particles are moving towards the concentration gradient. What is the meaning of concentration gradient? A gradient is in a graph. This slope of the graph is called gradient. So the concentration is high means the higher point of the slope, and the lower concentration means the lower part of the slope. So if the particles are at a higher concentration at one place and lower concentration at one place, they will swiftly, they will simply just, flow down. So we say they're going down the concentration gradient. The particles are moving down the concentration gradient. And since the particles are flowing down the concentration gradient, they do not need any energy. They do not need any energy. This is the reason that sometimes we can say diffusion is also passive transport. Diffusion is also called as passive transport. Why? Because this process does not require energy. Why this process does not require energy? Because the particles are moving down the concentration gradient. What is the meaning of down the concentration gradient? From high to low concentration, from the area they are more to the area where they are less. Diffusion takes place in both living and non-living things. In case of non-living thing, it is simply the spreading of the particles, the spreading of a fragrance or a spreading of the liquid. But in case of, living things, it is taking place across the semi-permeable cell membrane. So what does it mean? Diffusion is a process in which the particles can move in case of the living thing without cell membrane, and in case of the living thing with cell membrane. It means that cell membrane is not going to hamper the process of diffusion. Did you understand this thing, Hiba? So in our cells
Yeah.
also, in our cells also, how oxygen and nutrients are coming from the blood to our cells, it is just by the simple process of diffusion. Another fact about diffusion is, another important fact about diffusion is, another fact of the diffusion is, if the size of something, like size
Yes,
of
should I
the,
write this down?
you can write, or you will see the video recording later, isn't it?
Oh, yes, yes.
So when you will see the video recording today at night, see the video recording and then copy down
Then
all the
I,
important points. Okay. So if the size of the cell is extremely very small, microscopic, isn't it? The size of the cell. Such type of the things having a very small size have got a large surface area. So they have large surface area. The cells have large surface area, the area of their surface. And diffusion in such cases is fast, takes place very fast. If the size of something is very small, the surface area is very large, diffusion will take place faster. Opposite, if the size of something is larger, the surface area will be very small. The diffusion will not take place faster. For example, human body, you consider the example of human body with a small single cell. The size of the human body is huge. So the surface area of the human body is very small. If you will think that if I will not breathe through my nose and through my lungs, I will just simply diffuse oxygen from air across my skin into my body, into my cells. It will be impossible. Why? Because the surface area of the body, the large size of the body, the surface area is very small. So the diffusion, first of all, it is not possible. But if we even think that we can do that, the diffusion process will become so slow that we it will take long time for oxygen or nutrients from the surrounding across our skin getting inside the cell will take a very long time. So we will not be able to survive. This is the reason that we eat something, we breathe air, oxygen and nutrients from our lungs and from intestine, they go to the blood, and then blood take the molecules to the cells, and the cells are extremely very tiny, so they have got a very large surface area. So quickly when the molecules reach beside the cells, they will quickly diffuse inside, because larger surface area, faster diffusion. A small size, large surface area, fast diffusion. Big size, a small surface area, slow diffusion. Clear Hiba this idea? So are you there? Is it okay?
yes.
Have you understood? Acha. So how we are going to prove in our book, this is one small experiment is given. How we are going to prove that small things have large surface area and volume ratio. We call it surface area. Surface area is to volume ratio. Okay, surface area is to volume ratio. So if the size is very small, the surface area is to volume ratio will be very large. And if the size is very big something, the surface area to volume ratio is very small and diffusion will become slow. How we are going to prove that the smaller things have got large surface area to volume ratio? We are going to prove according to our book. We have got 3 cubes which are made up of agar gel. What is an agar gel? What is an agar gel? It is a gelatin, transparent gelatin, which has a consistency similar to our cell cytoplasm. So in biology, if we want to do the experiments related with cell cytoplasm, we use agar gel. So agar gel A cube, cube B, and cube C. So for example, this cube, each side is, let's say, 0.5 cm. Cube B, each side is 1 cm. Cube C, each side is 1.5 cm. So we have dissolved the gelatin in water and we have set it in different size of the molds. So the smallest cube, the larger cube, and the largest cube. Now we are going to find out the surface area to volume ratio of each cube. So let's start with cube A. Okay. So what is the formula of area? Length into breadth. All right. So 0.5 multiplied by 0.5. So what will be the area? Calculate. So the area of the smallest cube will be 0.5 multiplied by 0.5 will be 0.25. Okay, so 0.25 cm square is the area. So in order to find out the surface area, what you have to do? Because each cube has got 6 surfaces, so the area will be multiplied by 6. You get the surface area. So 0.25 multiplied by 6. So that will give you 1.5 cm square. So this is the surface area of cube A. Now, if we want to find out the volume of cube A, so volume will be length multiplied by breadth multiplied by height. So 0.5 into 0.5 into 0.5. Okay. So 0.5 into 0.5 into 0.5. 0.125. 0.125. Now I want to make the ratio. If I want to make the ratio, how I'm going to make the ratio? Ratio of the surface area to volume. So you will understand that what ratio comes for the smaller cube. So if I do it over here, I'm writing it in short, like if I do the ratio surface area by volume. So surface area is 1.5. Volume is 0.125. So if I divide it, 1.5 divided by 0.125, 12. The surface area to volume ratio of cube A is 12. Similarly, if we find out the surface area to volume ratio of cube B, how we are going to find out? This is cube A. Now cube B, area, length into breadth. 1 into 1. So 1 cm square. Surface area. What is surface area? Area multiplied by 6. So 1 multiplied by 6. So that is 6. So this is the surface area. What will be the volume? Length into breadth into height. So that is understood. Every side is 1. So that will be 1 into 1 into 1. So the volume will be 1. All right. Now cube B. If we want to find out the surface area to volume ratio of cube B, what it will be? Surface area by volume for cube B. 6 is the surface area. Volume is 1. 1 into 1 into 1. So that is 6. Now compare smallest cube, largest surface area to volume ratio. Larger cube, small surface area to volume ratio. Now the third one, this one. Okay, so what will be the surface area? So I am making the work shorter. Area into 6. So 1.5 into 1.5 multiplied by 6. So 1.5 into 1.5 is 2.25 multiplied by 6. So 13.5. Surface area is 13.5. And what will be the volume? 1.5 into 1.5 into 1.5. Length into breadth into height. 3.375. 3.375. Now let's do the ratio. So this is how we are going to prove that the size actually the surface area to volume ratio actually depends upon the size. So what will be surface area to volume ratio of cube C? So 13.5 divided by 3.375. So 13.5 divided by 3.375, 4. That is 4. Now see, smallest size of the cube, largest surface area to volume ratio. Largest size of the cube, smallest surface area to volume ratio. Now, let's think this cube A is a small cell This cube C is the human body. Its smallest size, larger surface area to volume ratio. Largest size, smallest surface area to volume ratio. So if the surface area to volume ratio is very large, diffusion will take place faster. If the surface area to volume ratio is small, diffusion will become slow. How we are going to prove this thing? So we are going to prove this thing. We are taking a big container. In the container, we have taken some colored solution. Okay. And in the colored solution, your 3 cubes, A, B, and C, you are going to dip for some time. This is made up of agar. So the colored solution has higher concentration outside. So the colored molecules, the colored solution will diffuse inside the gelatinous cube. Now, after some time, we will take the cubes out. We will cut the cubes in the middle, and what we are going to observe with a sharp knife, you will cut the cube, and you will see the smallest cube has become colored throughout inside, totally colored from inside. The larger one has become colored, but a small area is left in the middle. The color has not reached to the middle yet. Yes, if you will leave it for some more time, it might reach. It will reach, but it has not reached yet. All right, so the small portion is white inside. Now, the biggest cube, you will see only the color has reached up to the edges. Only the color has reached up to the edges in the same period of time. The diffusion has taken place faster in cube A because it has got a very large surface area to volume ratio. Diffusion is very fast. The colored solution has reached inside. In B, the size is a bit bigger. Surface area to volume ratio is a bit smaller. So diffusion has become a little bit slow. So the color has gone inside by diffusion, but a small portion is left. And the third one is the biggest cube with a smallest surface area to volume ratio with very slow process of diffusion. So the colored molecules have reached only up to the edges. So this is the proof that cells are microscopic, have a large surface area to volume ratio. Diffusion will take place faster. Nutrients and oxygen molecules quickly get inside the cell. Waste molecule quickly get out of the cell. But it is not possible in the large organisms like human beings. If we want to take the molecules from surrounding across our skin inside our cell, will be impossible because our surface area to volume ratio is so small that diffusion will become so slow that it will not help us to get the molecules by diffusion from surrounding into the body. But it is taking place on the cellular level. You have to send the molecules to your blood. The blood will take the molecules to your cells, and then the diffusion will become possible. Clear, Hiba, this idea?
Yes.
Acha. This is diffusion. Now, what is osmosis? What is osmosis? Osmosis is also a passive transport. What is the meaning of passive transport? Without any consumption of energy. Passive transport, without any consumption of energy, the molecules will move. What type of the molecules? In case of diffusion, we have seen all the liquids and gas molecules. They will move by diffusion in and out of the cell, all the liquids and gas. But in case of osmosis, only water molecules move. Only water molecules move. How? From the area they are more to the area they are less, or in other words, from high to low concentration or towards the concentration gradient. So that's why the molecules are moving from high to low concentration. They do not require any energy. So osmosis is also a diffusion, but it is the diffusion of only the water molecules. Why not the other molecules? Because there is something. The thing is that the molecules have to always pass across a semi-permeable membrane, no matter it is a living membrane or it is a non-living membrane. but there must be a semi-permeable membrane. If you will remove the semi-permeable membrane, it will not remain osmosis anymore. It will become simple diffusion. It will become simple diffusion. So in the cells, if there is water outside and the cytoplasm is more concentrated, less water, the water will get inside the cells in this way. Or you make it reverse. If the water is more inside the cell and less outside, the water will come out of the cells. So it means osmosis will take place only across the semi-permeable cell membrane, and due to which only the water molecules move from the area they are more to the area they are less. Now, suppose this is an animal cell. Just 1 minute, my writing device is... Okay, suppose this is an animal cell. This is an animal cell. Okay, so this is the animal cell, and you have placed it, it's our one of the red blood cells, let's say. You have placed it in a tray of water, in a container of water. The water is more outside, the water is less in the cytoplasm. Osmosis will start. Water molecules will start moving from high to low concentration across the semi-permeable cell membrane. As a result, there is a vacuole. A bubble of water will appear. This bubble of water will become bigger and bigger and bigger. Eventually, it will become so big that it will start pushing the cell. And as a result, after some time, the cell will burst and everything will be coming out. Why the cell is going to burst? Because there is no protective layer outside. There is no cell wall outside. There is no cell wall outside. It is made up of only cell membrane. Cell membrane is stretchable, but it has a limit. It will pierce, it will burst, it will tear apart, and the cell will die. You make it opposite. How we are going to make it opposite? Now, you have got a tray in which you have taken a very concentrated salt solution. In water, you have dissolved salt. Okay, and this is the cell. So water is, due to the presence of salt, water is less outside and water is more inside because you have put so much of salt outside. As a result, what will happen? The water is more inside, less outside. The water will start coming out of the cell. And after some time, the cell will shrink away and eventually it will die. So negative osmosis is taking place. Negative osmosis is the movement of the water molecules from inside the cell to the outside. So negative osmosis is called plasmolysis. All right. Positive means the water is coming inside the cell from outside. That is osmosis. Negative, water is going out of the cells because the concentration is changed. So negative osmosis, in which the cell is losing the water molecules, is called plasmolysis. This is the case of the animals. Now see the case of the plant cell. So this is the plant cell, and the plant cell has got a very rigid cell wall all around it. Okay. So there is a protective layer. Now we are dipping the plant cell in water. So water is more inside, outside, less inside. Osmosis will start, water will get inside. The vacuole will become bigger and bigger and bigger, but the cell is not going to burst. The cell will not burst. Rather, the cell will become inflated. The cell will become inflated like a balloon. The cell will become stretched like a balloon. The cell will become very fat like a balloon because of a big vacuole inside. Such type of the cell is called a turgid cell. What is a turgid cell? A turgid cell is a cell in which osmosis has taken place. The cell has gained the water. The cell has become very much turgid. The cell has become stretched, inflated like a balloon. So this cell is the turgid cell, and it is not going to burst because there is a cell wall around it. So osmosis plays an important role in plants. Osmosis plays a very important role in the plant. If osmosis takes place in our cells will burst. So it should not take place in our cells. But if it takes place in case of the plant cells, it is very much beneficial. Why? Because when every cell of the plant will become inflated like a balloon, the size of the plant will increase, growth will take place, and the plant will be able to stand erect. We have got a bony skeleton. We can stand. Plants do not have a bony skeleton. They cannot stand by themselves. They need an internal support. So from where the support is coming? Every cell, when absorb water, will have got a big vacuole full of water. Every cell will be stretched and inflated like a balloon. And so the plant will be able to stand erect. But if you make it reverse, like if you have dissolved so much of water in the surrounding, if you have dissolved so much of salt in the surrounding, so this is the salt solution, and you will dip the plant cell inside it. You have dipped the plant cell inside it. Okay, this is the plant cell. As a result, what will happen? The water is less outside, and water has become more out inside. So water will come out of the cell. As a result, what will happen? The cell will shrink away like this. The cell will shrink away. Cytoplasm will shrink away from the cell membrane. Such type of the cell is called a flaccid cell, which is due to the plasmolysis. Negative osmosis is taking place. Plasmolysis. So think if every cell of the plant, if it becomes flaccid, if it leaves, if the water molecules are going away because outside the water is less. So the water is drawn outside. All the cells will shrink away. All the cells will become flaccid. All the cells will become smaller. The plant will lose the internal support and the plant will not be able to stand. Rather, it will fall down. So because of plasmolysis, the cells will become flaccid and the plant will fall. This phenomena is called, in which the plant is falling down. The plant is not being able to stand erect. This phenomena is called wilting. The plants will wilt. The plants are going to wilt. Is clear, Hiba?
Yes.
Acha, let me show you something. Just 1 minute. All right. I'm sharing my screen with you. Look at this picture.
Okay.
Okay, now see this picture. This is one plant cell. It is surrounded by a salt solution. So you can see gradually, slowly, in between the cell wall and cytoplasm, gaps are created. Gaps are created. You know the reason why? Because water is moving out of this vacuole from high to low concentration. And then in cell number 3, the gaps are more. And then in cell number 4, you can see the cytoplasm is concentrated in one point, and so large gap is created in between the cell membrane and the cytoplasm. So this is the plasmolized cell. This is the plasmolized. This cell is the plasmolized cell. All right.
Okay.
Now see this picture. This is the picture I showed you for animals. Okay. So This picture. Have you seen? This is the red blood cell. The water is coming in. Outside, the water is more. Inside, the water is less. So water is going in by osmosis and the cell will burst. The cell is going to burst. So this is the thing. Now look at this. Water molecules are coming in. The cell is going to burst. The water molecules going out by plasmolysis. The cell will shrink away. The cell will shrink away. All right. Now this is another thing you can see. Yes. So this is the onion cells. So if you will take the onion tissues and if you stain them and if you observe them under microscope, you will see these brick-shaped cells of the onion. So if we put some salt solution over the onion cells, so water start coming out of the cells by osmosis, and you can see how the cytoplasm has been shrinking away. Gaps are created. These are the cells which are turgid. Water is there. But gradually, slowly, the cells are losing the water and gaps are created between the cell membrane and the cytoplasm. Got it, Hiba?
Okay. Yes.
Acha. So this is all about osmosis and plasmolysis. So what is the osmosis and diffusion? So diffusion and osmosis have got some similarities. What similarities? In both of them, the molecules, both of them are the movement of the molecules. So when you will watch the recorded video, you will write down these points. Both of them are the movement of the water molecules. Number two, in both of them, the molecules are moving from high to low concentration. Number three, in both of them, energy is not needed. Energy is not required. Why? Because the molecules are moving towards the concentration gradient. What does it mean? From high to low concentration. From high to low concentration. In both of them, the molecules can travel over a large distance. The molecules can move over a large distance in both of them. Okay, the molecules can move over a very large distance in both of them. And what is the difference in between osmosis and diffusion? The difference is that diffusion is diffusion is the movement of any sort of liquid and gas molecules. But osmosis is the movement of only the water molecules. And in case of diffusion, it can take place with or without semi-permeable cell membrane. But osmosis takes place only in the presence of cell membrane, living or non-living, but the cell membrane must be there. What is osmosis? Positive osmosis, in which the water molecules are getting inside the cell, is osmosis. Negative osmosis, in which the water molecules are going out of the cells, is plasmolysis. So if the cell is surrounded by water, water will get inside. Osmosis will take place. If the cell is surrounded by a very concentrated salt solution or sugar solution, then the water will come from inside to the outside. The cell will shrink away. The cells will die. That is called plasmolysis. Osmosis causes turgidity. The cell will become inflated. Plasmolysis causes flaccid cells, flaccidity. The cell will shrink away. Clear, Hiba?
Yes.
We have got 5 minutes left. In 5 minutes, let's finish another small thing, that is active transport. Today is Friday, actually, so we have only, like, means I have only 1 day of holiday. Again, from tomorrow, my marathon will start. So that's why I will be taking only 1 year, because I have to go out. I will finish your class and then I will just go out for some shopping. So I will quickly finish active transport, and then today at night, you will watch the thing. I will give you the slide and notes, and you will start your preparation. You have a test very soon. Okay. Acha. What is active transport? Active transport is the movement of the molecules. What type of the molecules? Selective ions. glucose molecules, amino acids molecules, salts, etc., etc. How? It's opposite from low to high concentration. What is the meaning of low to high concentration? From the area the particles are less, will go to the area where the particles are more. What does it mean? Against the concentration gradient? Against the concentration gradient. Against the gradient, ulta. Low to high. And this is the reason that the molecules are actually climbing up to the height. So it requires energy. Where is the energy? Only in the living cells. It means active transport is taking place only in the living animal and plant cells. Only in the living animal and plant cell, because energy is needed in this process. Dead cells do not have energy. And always across a living cell membrane. Living cells have living cell membrane. So from where the energy is coming, actually, cells have got mitochondria. Mitochondria have got energy in the form of ATP. So the cells are actually using this energy to pull the molecules inside from low to high concentration. So in our cells also, glucose, amino acid, and salts are getting inside our cells by active transport, and in case of plants as well. From the soil, the salts and other minerals are getting inside the roots by active transport. So this process requires energy.
Okay.
So this is all about the osmosis, diffusion, and active transport. So we have finished chapter number 1. All right. So in the next class, we will try to finish chapter number 2 in one class, because chapter number 2 is a small chapter about the microorganism, fungi, bacteria, and virus. So we will finish it in one class. So our chapter number 1, 2 will be finished. Digestion and respiration is finished. Will be finished. All right, Hiba. So that's all for today. I'm uploading the class recording and the slide right now. So please sit and study everything. Okay. Allah Hafiz. Bye.